Hello, my name is Matthew Busse. I'm a senior curator working on the CEDAR database, and today we'll be going through a brief overview of the curation process. Curation is the process of transforming unstructured data from the scientific literature into structured, standardized, searchable data presented in the CEDAR database. So papers of interest are identified from the scientific literature. Then a team of curators extracts, formats, and enters the data into the database, resulting in a single resource of collated, standardized data, which allows users to search through the database for data relevant to their own research. Let's look a little more closely at the first step of identifying relevant papers. This is done using a custom-made biweekly automated query that scans through PubMed every 2 weeks, looking for papers of interest to the database. Those papers are then sent to an automated text classifier, which sorts through at a more granular level, looking for uncuratable papers that are lacking critical information. The remaining papers are then grouped into categories. Then the next step is a senior curator scans through the abstracts of those papers to provide human-level confirmation that the papers are actually curatable. And then once confirmed, those papers are assigned individually to curators for manual curation. And we'll be looking at an example paper today to help bring the curation process to life. So you can see here the PubMed ID of this paper. This is a cancer paper that I recently curated and is now available in the CEDAR database. So now let's look more closely at the manual curation step. So the first step is for a curator to read through the paper, looking for epitopes that meet the minimal inclusion criteria for CEDAR. Those criteria are, it must be either a linear peptide smaller than 50 amino acids in length, or a discontinuous epitope containing individual residues that have been shown to be critical for recognition. And additionally, in order to be curatable, an epitope must be tested in an assay as either an immunogen or an antigen, and we'll go over specific definitions of these in a little bit. And additionally, there is other minimal information required, such as the exact sequence of the epitope. And then for the assays, the epitope is used in other information such as the host and outcome is also required. So looking at our example paper, you can see here an epitope sequence is provided, as well as curatable assays. So this data is entered into the database as structured data. So a big part of our job is mapping the data from the literature to the data structure of our database. So this is a simplified overview of the data structure. And as you can see, the data are organized around epitopes and assays. So a curatable paper will have one or more epitopes within it, and each of those epitopes will be tested in one or more assays. And it's the assays are where the meat of the data are captured. And we'll go through a few key definitions now. So the host of an assay is the species that is producing an immune response. The immunization is the event which induces an immune response. And this can either be the occurrence of a disease, such as cancer, or the administration of an immunogen in a lab to induce a response. Another key field is the antibody or effector cells, and these represent the mechanism of the immune response. Then finally, the antigen of an assay is the entity which is recognized by the immune response. So in our usage, an immunogen is what induces an immune response, and an antigen is what is recognized by the immune response. And for completeness, our definition of an epitope is the specific molecular structure interacting with the immune system. So now let's look more closely at data entry. And this is done using our custom-built in-house data entry portal. You can see here an example, a screenshot of the Modify Epitope screen. And here you can see the epitope sequence has been entered from our example paper. And each peptidic epitope that's curated is linked to a source ID of a protein in GenBank or SwissProt. So this links all peptidic epitopes to known existing proteins. And now let's look at an example of an assay data entry. So here's an example figure from our paper. It's a dextramer assay. And here's a screenshot of the modified T cell assay screen. The assays are broken up into T cells, B cells, and MHC binding assays because they each have some unique fields specific to that disease, to that assay type. But there are several common fields in all of them. So here you can see the host of this assay is human. The immunization is occurrence of cancer, in this case, metastatic melanoma. Here you can see the assay information has been entered along with the outcome of the assay. And here are the effector cells, in this case, CD3 positive T cells from blood. And here the antigen of this assay is the epitope itself. Whenever possible, data are entered using controlled value fields. This means that the descriptor of the data is selected from a drop-down list of standardized terms. And this goes a long way towards ensuring that data are captured consistently from paper to paper and curator to curator. However, there are some limited free text comment fields, and these are necessary to help us accommodate any extra details or unique situations which are not easily fit into controlled value fields. And it's this balance of controlled value fields and free text fields that enable us to consistently capture data while still allowing for the flexibility needed to accommodate the creativity of the scientific process. One tool that helps us do our job is the Curation Manual. And this is an online wiki of tools that govern how data are mapped to the data structure. We have a general curation manual, and then also specific curation manuals for different diseases that describe how to handle unique situations that arise in those diseases. And this is a living document that's updated as needed as we encounter new situations that require descriptions. And this is available publicly if anyone is curious. Another tool that we have to help us do our job is a series of ongoing field level data validation checks. So several fields have automated validation checks that run once data are entered into the field. So for example, when a peptide sequence is assigned to a protein ID, if that peptide sequence is not actually present in the protein ID, it raises this error message, which warns the curator that the peptide sequence is not found in the protein and a different ID is needed. And once the curator finishes the initial curation of the paper and the curation are then sent to another team member for internal peer review. And so the reviewer looks over the paper, looks over the curation, and provides human level confirmation that the data are mapped correctly to the data structure. There's too much variability to rely solely on automated checks, so we do include this additional step of a human confirmation. And this can be an iterative process with rounds of back and forth between the reviewer and the curator as necessary. And then when the reviewer signs off on the paper, there's a final level of curation level data validation checks. So these are automated checks that look at a higher level to ensure that the data are mapped correctly to the data structure, looking at things like relationships between epitopes and assays. So if an epitope is entered but not assigned to an assay, that raises an error message. And once those validation checks are complete, then the curation is promoted and made publicly available on the CEDAR database. So searching through the database for the PubMed ID of our example paper finds one epitope with 3 assays. You can see the assays here all linked to the same epitope. And Our multimer tetramer assay is present as well. And so data are published to the database weekly. And since we formed CEDAR in 2021, the teams focused on and curated 2,350 papers. As Alex mentioned earlier, we curate the current year papers, 2025, as they're identified in our biweekly automated query. And then we're going backwards through time, curating the existing papers. And currently we're working on 2013. So that is a brief overview of the curation process. I'd like to thank you for your time. And now we will take questions.
Thank you so much, Matt, for that wonderful presentation. While Matt takes a look at the Q&A, I'd like to invite Alex back to the virtual stage to take the first question. And in the meantime, I'll be launching a poll in the webinar as well. So thank you, Alex.
Yes, so there is a question whether we are using ChatGPT for curation, and the answer is no. We have a strong interest in developing AI-assisted methods for curation, and we're working on it. But at the present time, the AI methods just don't have the accuracy that we desire and demand from our data. So we really need to do this at this point in time manually. But in the future, we might benefit from using AI. Matt, do you want to take the how long does it take to curate a paper question?
Sure. Right. So we have a question. How long does it take to curate one paper? Who are the people doing the data entry? So that it really varies. It depends on the length and complexity of the paper. One paper can take anywhere from 1 hour to 20 hours for large, very complex papers. It greatly depends on the number of epitopes and the number of assays that are in the paper. Then the people doing the data entry are all PhD trained scientists, such as myself, mostly who have some kind of background in immunology or cancer biology.
So I will take the next questions. There is a question that says, I generated a data set of cancer epitopes. How can I make them to be included in CEDAR? Well, there are two paths, so to speak. One is get your data published in a peer-reviewed journal. That is a very good thing because it does provide an additional quality control, so to speak. Short of that, in certain situations, we have included data sets that have been generated, for example, in the context of the IDB by NIH-sponsored contracts and so forth. And that also has been a source of very interesting and good data. And the inclusion of not yet or not published data is done on a case-by-case level. And so if you do have a data set, please feel free to contact us and we can take that discussion offline. Matt, back to you. What did you pick?
All right, we have a question. Are you curating tumor proteins of different organisms? Right now, and correct me if I'm wrong, Alex and Nina, we're focusing on human cancer at the moment. We do include studies from animals if there are also human epitopes included in the paper, but at the time, due to, I think, resource constraints, we're focusing solely on human epitopes.
Whoops, I'm sorry. There's a question is what tools are you using to run the initial complex query? So the initial complex query is twofold. One is just a simple PubMed, very, very comprehensive query with lots of keywords and so forth. And then following that, that is further analyzed by a in-house developed classifier application, and that has been published in a specific paper describing this text recognition application.
All right. Here's a question. At the end of Mr. Busse's presentation, it was mentioned that papers from 2013 are being curated right now. The data set as now contains information from before or after 2013. Oh yeah, sorry if that wasn't clear. So right now we have curated from 2013 through 2025. And we are working backwards from 2013 to earlier. So right now in the database is 2013 through 2025.
So there is a question that says, great presentation. Thank you. You mentioned patterns can be developed from epitope info in the database. How does CEDAR balance those more collaborative shared values with privatization and patents? Are there discussions around moving towards less proprietary and encouraging more open access tools and products instead? Well, I mean, our goal for CEDAR DIDB is to catalyze progress, whether that's basic research or applied research. We are equally pleased of progress in those two dimensions. The data is all it's all open and generally accessible to the scientific community, whether you are a basic scientist or work in industry. So we don't balance in the sense that it's all open access. Then whether you use the data or the tools to develop something that is innovative and patentable, actually, you could be a basic research in a university that is making a discovery that is then patented by university. So it's all open and in the open. Companies also like to license tools, we found, because they, if they have a secret protein that they're working on, they want to be able to run the tools without necessarily making the information of what is the sequence of a protein we're working on spyable online. But that's a different issue.
All right. Another question here. If you find a neoepitope or in a paper, how do you map it in the reported protein? That's a great question. So when we find a neoepitope, we use protein blast from NCBI to map that to a wild type sequence, because blast allows for mismatches. So that helps us identify if it's a neoepitope that has a single substitution and still in frame, then we can map it easily to the corresponding wild type protein. And there's a field in the database for when we curate epitopes, we can add an epitope-related object, which we use to curate neoepitopes so they are linked to the wild-type epitope if one can be identified. Or if it's a neoepitope that greatly changes the structure, like an indel, then we can map it to the protein, just to the protein itself. But so yeah, we use BLAST to map neoepitopes to the corresponding wild-type epitope, and they are linked in the database.
There is a question whether the CEDAR citation index excludes self-citation. Yes, it does.
And while Matt is picking his next question, we are going a little bit over time, which is totally okay. There are a few more questions to answer. I would just like to remind everyone to please post your questions in the Q&A rather than the chat, just to help us manage them. But Matt, we'll go for another couple of minutes if you'd like to pick the next one.
Okay. Here's a question. Are you curating autoantigens and epitopes? Will your database identify the overlapping epitopes between cancer and autoimmunity? That's a complicated question because we curate data linked to a disease, and if two disease processes are involved with an epitope, such as cancer and an autoimmune disease, or cancer and an infectious disease, we do have a way to link in the assays to capture both of those disease processes. We don't outright identify an epitope as belonging to cancer or belonging to an autoimmunity or infectious disease because there is often overlap. We can't always say an epitope is only involved in one type of disease. So we do curate multiple disease contexts, and I hope that answers the question.
Okay, I suggest, in order to be respectful of the other presenters, that we could probably end the Q&A now, if Nina is in agreement as well.